Hey everyone, my name is Stephen, and in this presentation, I'm going to talk about the pro-inflammatory role of platelet signaling in rheumatoid arthritis. I work for Dr. Hugh Kim, and we study platelets very extensively. So first of all, rheumatoid arthritis is a very prevalent autoimmune disease. It causes joint degradation, disability, and cardiovascular diseases. And it causes lymphoma and other malignancies because of the intense inflammation it results. Two cell types are very important for the pathogenesis of this disease. The first one is fibroblast-like synoviocytes, and the second one is platelets. So in rheumatoid arthritis patients, there's an elevated level of circulating cytokines. These make platelets more prone to activation, and then they secrete pro-inflammatory and prothrombotic mediators, including platelet factor 4, and they secrete interleukin-1 containing microparticles. These cytokines and interleukin-1 containing microparticles, they travel to the joint and activate a cell type called fibroblast-like synoviocytes. Normally, they live in this layer right here called the synovial membrane, and then they secrete matrix metalloproteinases and different cytokines to cause immune activation and joint degradation. So my research question is that, does platelet factor 4 specifically induce a destructive phenotype in fibroblast-like synoviocytes? The first question I want to answer is that, does fibroblast-like synoviocytes express platelet factor 4 specific receptors? Platelet factor 4 binds to CXCR3A, CXCR3B, and CCR1, and some other receptors that we don't know the protein sequence of. So I'm working with what I can here, and I can only study whether fibroblast-like synoviocytes express CXCR3A, 3B, and CCR1. I decided to study this phenomenon using a bioinformatic approach. Some other group has already sequenced the transcriptome of fibroblast-like synoviocytes, and I downloaded the FAST2 files from NCBI sequence read archive, and I searched the sequence of CCR1, CXCR3A, and CXCR3B within the transcriptome using Magic Blast. I found sequences related to all three receptors inside the transcriptome of fibroblast-like synoviocytes. That means platelet factor 4 probably has an effect on fibroblast-like synoviocytes. In the future, I have two future directions. So I will co-culture a cell line of fibroblast-like synoviocytes called K4IM cells with platelet factor 4. I will study the cytokines secreted and the matrix metalloproteinase secreted using both ELISA and quantitative PCR. I will also study the proliferation rate of the cell line using EDU proliferation assay, because fibroblast-like synoviocytes, they proliferate at an elevated rate in rheumatoid arthritis. I will also study the intracellular signaling pathway from the binding of platelet factor 4 to their receptors, which leads to the secretion of matrix metalloproteinases and cytokines. I would like to thank my supervisor, Dr. Hugh Kim, and my fellow lab members for helping me along the way. And I would also like to thank CBR for making this research possible. With that, I'll take questions.
Stephen, do you have any idea as to how anti-TNF therapy or drugs might affect PF4 secretion?
Anti-TNF, you said?
I mean, because those are the drugs, my understanding, I mean, that's the type of drug that is generally used in patients, or one of the drugs that's used in patients.
That's a very good question. I don't think anybody has studied this effect, so that may be one of the things I can measure.
How will you, we're good. How will you study binding of PF4 without inducing cellular changes that might induce new binding sites?
Why do I need to study new binding sites?
New binding sites. So I'm wondering how you will avoid, will you be studying the intrinsic binding or will you be studying induced binding? So how will you be able to distinguish between those two events?
So as far as I know, platelet factor 4 binds to, platelet factor 4 binds to the receptors, so CXCR3A, 3B, and CCR1, and these will induce an effect on the cells that have these receptors. I'm not quite sure about like new binding sites and other things you were talking about. Would you mind elaborating on that a little bit further?
We can talk later.
Thank you.